Good morning, everybody. Welcome to the webinar. We'll get started in about 1 minute as attendees trickle in here. I wanted to make a few announcements. So this is the NSF RCN webinar on SARS-CoV-2 RNA and wastewater. And the theme of this webinar is use cases and public health use of wastewater data. A few housekeeping items during the webinar. You should put questions into the Q&A, not into the chat. The Q&A offers an upvote option. So if you're looking at the Q&A and there's a question that you'd really like to see asked of the speaker, then you can upvote that question, and the more upvotes it gets, the higher the priority it will be during the Q&A session. I'm going to go ahead and introduce our speakers. So today we have Nathan LaCross, who's the program manager at Utah Department of Health, and then Loren Hopkins, who's the chief environmental officer at the Houston Health Department. Nathan is going to be speaking first. He has a 20-minute presentation and we'll have 10 minutes for Q&A. Thank you so much to Nathan and Loren for joining this webinar to share their work with us. So with that, Nathan, please go ahead and share your screen and you can get started. Thank you.
Sounds good. All right, hopefully you can see my slides once again.
Yes. Yes. Yes.
Excellent. All right, thank you so much for that very kind introduction, and thank you so much for having me and giving me a chance to speak to you. Very pleased to do so. I think that there's a lot of really interesting things to talk about with wastewater surveillance or wastewater-based epidemiology. I kind of use them interchangeably. As Ali mentioned, I am the program manager for the wastewater surveillance program at the Utah Department of Health, which, as you might imagine, is quite new. This being a relatively new field, most everywhere in the country, and is rapidly expanding. So as I mentioned, lots of areas and opportunities for discussion and for research and for just gaining new valuable knowledge. And I'm going to talk, that's really going to be most of what I talk about. Talk a little bit about what we've done in our program in Utah, because we were a fairly early adopter of the idea. But mostly what I want to talk about are what are some of the open questions and what are some of the important things that we really would like and need to know to keep moving forward with this. So there we go. So very briefly, with our wastewater surveillance program, we started back in April of 2020. The planning started back in probably mid-March of 2020, so quite early on in the pandemic. And it was really just the result of a very, very fortunate confluence of our very active and good partner in our Department of Environmental Quality, some excellent researchers at a few of our universities. You can see them there: University of Utah, Utah State University, and Brigham Young University. And they were looking for an opportunity and a way to do something interesting, but really to help the state's pandemic effort as well. Not being directly involved in public health as we were the health department, of course, that wasn't their role, but they still wanted to do something. And they had some experience looking in wastewater for other things, and this seemed to be a good opportunity to try it out on a new target, SARS-CoV-2 in this case. So it started with a relatively small pilot study, about 10 sites, which they were fairly large wastewater facilities. So they did cover about 40% of the state's population. Sampling once a week. That was chosen because we didn't know what else to do. There was really no guidelines. We were kind of making up as we went along. But we did know, of course, that it was very important. We're going to have 3 different labs analyzing these samples in 3 different locations. So it's very important to have some uniformity in methods there. And they spent quite a bit of time and doing things like split samples and sending samples back and forth to make sure that the results were at least broadly comparable at all 3 labs. And this actually worked very well, possibly better than anybody was expecting. This kind of pilot phase went through the end of May 2020, and it worked well enough that we were all able to get additional funding from our what's called Unified Command here. So that was the multi-agency incident response command center that was overseeing the state's pandemic response, and as well as being responsible for distributing COVID-19 funds. And it had a lot of promise and showed some good results. And so we were able to keep it going and expand greatly. So kept sampling it once a week, but we expanded up to a maximum of 47 sites. This was probably by late summer, maybe early fall of 2020. And that ended up covering about 90% of the state's population. So just about everybody we were sampling in one way or the other about once a week. And of course, being one of the very early adopters of this, as I mentioned, really just as a result of fortunate circumstances for us, we were one of the initial members of CDC's National Wastewater Surveillance System, which has also expanded greatly in recent months, I think, which is fantastic. But one thing was becoming very clear as we were getting this data and trying to figure out how we can use it, and how our other public health partners, like our local health departments, could make use of the data, is that it really ideally needed to be more timely and more frequent than sampling once a week. Depending on delays at the labs, we might not get a result back for close to another week or so. And so ended up being sometimes a fairly significant lag between when a sample was taken, so when it was fresh, and when the result was actually available to public health practitioners. And that did raise some questions about, well, okay, that was what was happening last week, but what's going on now? So we ended up starting sampling more frequently. We changed it to twice a week, which was we felt a reasonable compromise between what we would ideally like in the, ignoring the practical considerations in public health. We really want just as much data as we can possibly get, as often and as timely as we can possibly get it. But it's not cheap to get samples in a very rapid fashion from across the state. And it becomes more and more of a burden on the partner facilities themselves, who often have, especially in the small sites, one or two people working, and sometimes only a few days a week at the wastewater facility. But even going to just twice a week, there's a number of facilities that just weren't able to cope with that level of sampling, unfortunately, and they had to drop out of the program. And that's most of what we ended up cutting. As an unfortunate side effect of that was mostly smaller sites in more rural areas, which would be of great interest to get samples from, because they often also have lower access to things like clinical testing. But we just couldn't make it work, unfortunately, so we had to cut them and drop it down. I think that the gain in the timeliness of the data that we're getting, moving to twice a week, more than made up for that, but it is an unfortunate thing. And of course, we also just had, funds are limited, resources are limited, so we can't sample everywhere all the time as much as we would like to. So back in January of this past year, we switched to twice a week sampling, and it dropped down to 33 facilities. As I mentioned, most of the ones that were dropped were some of the smaller ones that served smaller populations, and so that's still about 88% of the state's population that we're sampling. And you can see the map there. This is where we're currently sampling twice a week. Very, very briefly in the lab analyses, there's not, I'll get into this a little bit more later on in the presentation, there's not really a unified set of best practices anywhere in this field, unfortunately. Most people, I think, are doing something very similar to us, which is real-time qPCR assays. Other people are using digital droplet PCRs if they have the equipment available. We're assaying for the N1 and N2 loci. Most people are using at least one, sometimes both of those. There are occasional other targets you'll see. And these days, we're typically getting results in, quality checked and out and available to anybody who wants to use it or see it, typically within 2 to 4 days after sample collection, depending on whether there's a weekend intervening, which I think is really pretty good. That's pretty fast. And all the analyses were moved from our partner academic labs to the State Public Health Lab this past August. Our academic partners were absolutely fantastic and integral, absolutely key to the development and expansion of this program and making it as successful as it actually is. But as you might imagine, they wanted to get back to their research too. This is not what they want to do forever and ever, which I think is a very reasonable point of view. And really, probably the State Public Health Lab is a better home for an ongoing state-level surveillance program anyway. We're working on sequencing, don't have it quite working yet. I just got back yesterday from long vacation, so maybe they did it. I don't know, I haven't heard yet. Something I'm gonna investigate in the coming weeks, days, sooner than weeks. We have a couple of different dashboards where our data is available. This is just a screenshot of it where we show a map of that sewer shed. Hopefully that part is pretty obvious. The top graph in purple there, that is the wastewater data itself expressed in millions of gene copies per person per day. So basically kind of normalizes both for the population as well as, in there we're also normalizing for flow. There's a number of different ways you can express the data. I don't know that any one is more correct or better than the other one. At least I don't think there's enough information out there to definitively determine that yet. This is something that we settled on. It ended up being pretty similar to what CDC decided on as well. And in the bottom in blue is the daily case rate per 100,000 within that exact same geographic area. So we geocode all of our cases, and these are the ones that reside within this particular sewer shed as well. So this is kind of the comparison point. You see the wastewater data, compare it to the case data. So very briefly again, one of the, I think, most widely used and most useful aspects of wastewater surveillance data in this context is the early detection of rising infection rates. Wastewater data is often, not always, but often a leading indicator of things that are coming down the line in cases that we'll see in clinical testing. And you can see an example of this here. This is in fact one of the first, if not the first, real piece of evidence we had that this method was probably going to work pretty well, at least at times, for this. This was back in May of 2020, so really quite early data. And this is from Logan, a city in northern Utah, where Utah State University is. And you can see in the top, in purple, that kind of its circle, there's a sharp rise in the wastewater data. And again, we're sampling about once a week here. So that hence the kind of wider gaps between the data points. We see a really distinct and sharp rise in that. And then just a little bit later, we see a corresponding rise in the case rate within that same area. That really tall spike you see is because of a single very, very large testing event. You know, one of the downsides of our normal case data is very dependent on all aspects of that clinical testing. And so when you have a single large event like that, that can really influence how the data looks. And this is what we call case date. So it's a combination of a number of different date variables, but it comes down to roughly when the first positive result for that individual was first recorded. Onset date of symptoms would be better, but I have found through looking at it that it is so lagged and unreliable as to be essentially unusable. I hadn't looked in a while, but when I did, there were significant changes, 5, 10, 15% easily a month down the line, a month after, which is just, it's too variable, it's too lagged behind to really be useful, unfortunately, because it would smooth out some of these spikiness we see in the case rates. But you can see hopefully that we saw a very similar pattern in the case rates as we saw in the wastewater data, and the wastewater data that change preceded the similar change in the case rates. Another essentially the same type of example, but from much later. This is from September of this year in another city in northern Utah. These are representatives, so as we all know what that means, I picked the ones that look the prettiest and best exemplified the points I'm trying to make here. But this is a very common sort of thing you see with this type of data. So again, we can see that rise in levels in the wastewater data, and then a bit later, often the amount of lead time you get really does vary. Probably, you know, maybe 7 days, maybe on average a week, but it does, you know, I've seen as much as 2 or 3 weeks, and as little as nothing at all. It really depends on many different factors, but you can see a very similar pattern there as well. Another very useful aspect of this data, and I'll get into kind of the idea behind this a little bit more later as well, is you can use it to help confirm and add context to other surveillance data, like the confirmation of declining case rates. So back last summer of 2020, you can see in the top graph, or excuse me, the bottom graph, that is the case rate within that sewer shed, you see the map of theirs that covers Salt Lake City. And you can see declining case rates, which is great. And we saw very similar things across the county, in fact, across the state. This is after that first surge we had in summer 2020 that we thought was so bad, and we've learned better since. However, we also saw declining testing volumes within the county. You can see that on the top graph. That's for the entire county, but it was essentially the same sort of pattern for all the individual sewer sheds within this county, as well as other places in the state. So that raised the possibility that the declining case rates we saw were really possibly no more than an artifact of the fact that we just weren't testing as many people. But we were able to use wastewater surveillance data which decreased and then stayed low, indicating there were not nearly as many people shedding the virus into the sewage, to help confirm the idea that this was in fact probably a real decrease in cases. There probably weren't as many cases, and it didn't have really anything to do with the fact that we just weren't testing as many people anymore. And that's some of the main uses for the data. We've used it extensively to help identify where to send, especially mobile test teams, but also where to set up new fixed sites for clinical testing. Been using it for that since probably January or so. Used it to help identify where we should send masking observers. We did that for a little while. Targeting of outreach, education, other interventions along that sort of line. Let's skip forward just a little bit. Okay, here's more what I want to actually talk about. I took longer than I had anticipated before. So strengths and weaknesses, and a couple of barriers, first off, of wastewater-based epidemiology. So barrier-wise, I think there can be something like an inertia against working with really probably any new data source and new method, this one included. For some people, it's new, and so it's not fully trustworthy. No matter how well it seems to work, no matter how sound the logic and the science behind it is. Sometimes only time can overcome that. I think examples of where it is successfully predicted changes in their more traditional data sources also help greatly. And crises like the pandemic can provide rationale and impetus for spending money, time, resources, et cetera, on new methods like wastewater-based epidemiology. But because you're in a crisis, a lot of times these things really have to prove their worth very rapidly. Otherwise, they'll get passed by as it's not helping me right now, and therefore I don't care about it because I have way too many other things on my plate, which is an understandable point of view to some extent. So some strengths of this. One of the biggest ones I wanted to mention first was that it is in fact independent of traditional epi data. It's independent of healthcare seeking behaviors. It's independent of access to clinical testing. This is incredibly valuable. We know that our traditional data sources from that clinical testing are highly dependent on a person's ability to get tested based on what testing availability they have in their area, as well as their willingness to get tested. That's been a big issue lately, not just in Utah. Wastewater data, unless they are not in the system, so on septic tank, which is relatively rare in Utah, more common in other areas, but relatively rare here, or they just somehow don't make use of their municipal sewage system, we're going to get data from them. It's anonymous data. It's inherently a pooled sample, which is another strength, I think, at times. But it's independent of all those other considerations, which make it really, really useful, especially in situations where we're having trouble getting a good representative sample of people tested by traditional means. It captures all people who are shedding, not just symptomatic cases. So it will capture people who are asymptomatic or have mild symptoms, just as well as anybody who has full-blown COVID-19. As I mentioned, we'll often get early detection, well, not always. It's quite efficient. Expenses do add up, but we're sampling right now about 88% of the state's population twice every week for really probably pennies a person, which is pretty impressive when you think about it in that way. I try to make that point to often justify myself. As I mentioned, it's inherently a pooled sample, which can come in handy sometimes. There are really no, except for very small or institutional level sampling, there's really, I don't feel many privacy or data security concerns around it. We have no ability to tell precisely where aspects of the signal we detect are from within the sewer shed. We just know that it's in that entire area. That's all we know, and that's all we can get from that sample. And it can be used for lots of different targets. We have, I think, a number of different plans for the futures. You know, influenza. There are chemical targets like opiates. Lots of different things you can all use with the same sample. Really, you have to treat it differently when it gets to the lab, but it's the same sample. Some weaknesses. Lots and lots and lots of unknowns. So lots of opportunities for research, I think, and very exciting things, but there's a lot of stuff we don't know that we'd really, really like to. We often see fairly high variability that could well be due, at least in part, to some of these things that we just don't have information about yet, those unknowns. But we often see that high variability. As I mentioned, it's new, so it's not always fully trusted by some folks. I also mentioned, really best practices or gold standard methods haven't really emerged as of yet. I think we're moving there, but we're not there yet. And it'd be very helpful, especially when we're trying to compare results from different areas. If not even the methods are the same, it becomes very difficult. And based on all those unknowns, different methods, et cetera, it's really probably not very valid to compare data between facilities or much less different labs or different states. Which again is not a great thing. You like to compare data, and that's very difficult with this, I think. And ideally, because you can't compare levels that you're seeing currently to another facility or to some other metric or standard, really, because there's so many different things between each site. You really need some baseline levels where cases are low, ideally, for comparison purposes. So what you're looking for is changes over time within a facility. So research questions. Oh, I'm at 1954. Very, very quickly. So lots and lots and lots and lots. Here's all the things that I could think of off the top of my head that we don't know that I think could be very useful. Differences in sewer system infrastructure. I mean, how much does infiltration of runoff and groundwater come into play? Temperature of the groundwater, is that important to know? Probably, but we don't know it. And so there's lots and lots of different things here, lots of things that could affect the data that we see, the results that we see, that we don't have good information on, at least not regularly. Which ones are important to know? Which ones are the most important? And which ones do we need to take into account? How should we best take it into account? What are these best practices that everybody should ideally be doing, or maybe a set of them based on expertise and equipment availability, et cetera? And then these are just the known unknowns, right? What are the unknown unknowns, the ones I didn't think of? Those are probably important too. I just didn't think of them, hence they're unknown. So I think there's lots and lots of room there. Sorry to skip through that really fast. I'm happy to answer any questions you may have. Also, feel free to contact me at any point. There's our public dashboard if you ever want to see our data. Thank you so much. Let's see, Q&A. I had that.
Thanks so much, Nathan. This is great. It's really exciting to see how quickly you got on in Utah with this and how rapidly you were able to expand, as well as how you could transition this over from the universities to the state. So let's see, we have a number of questions here. Maybe let's start with one that had 4 upvotes here. This is Colleen Norton asking about the transition from the academic labs that early led this endeavor to then the state lab. Did you validate the methods? How was this handover being handled, and what's your experience and recommendation from that?
Yeah, that's a great question. And we did. We had an overlap of, a number of months where we knew we were going to be making this change to the state public health lab for a variety of reasons, in no small part, as I mentioned, because the research folks wanted to get back to their actual jobs, the research. And so one of the things we took a lot of care to do was not make sure the methods were identical, because that's, I think, less important. But we did quite a few different split samples to where basically one of our established labs would be analyzing them, and then part of the sample would go off to the state public health lab, and they would analyze it with the method that they were validating and trialing. And we'd compare it and see, like, what do the results look like? Are they comparable? Are they way different? In part because of that experience, as well as the fact that the efficiency of the method they were trialing just was poor. They ended up scrapping the method they thought they were going to use and went with something else. That is broadly similar to what our academic partners were using, but different also in a number of really important ways. And for the most, it ended up being interesting. It's variable how comparable the results are. For about half of the sites, and it does seem to go by lab, about half the sites, the results, you couldn't even tell without looking at the dates from each data point where the switch occurred, where it went from the academic partner and then to the state public health lab. In other ones, there was a big change, usually a large increase in wastewater levels. And as I mentioned, that was different by lab. And so I suspect that despite all the time and effort we spent doing interlab comparisons early on, that the methods had drifted over time. And so I think, and you know, especially with the academic partners, there were a number of students doing this, and they were great. I remember being a student myself. You know, they did great work, but they're still learning in a lot of ways. And so I think there's more variability in the results you get from student A versus student B, versus somebody who might have more experience there. So I think those are some really important things to keep in mind. Do a lot of comparisons, get a sense for whether the data is comparable or not. If it's not, doesn't mean that you shouldn't go forward, but it is something you're going to have to think about and take into account when you're figuring out how to use the data, and whether you should still mark off that old data in some fashion. It's like, hey, I don't think this is really comparable to what we're doing now.
Yeah, I think we did answer Aaron Viven's question. So maybe there was another one in the general discussion in the chat about the dashboard. Maybe let's explore this a little bit. So the one question was about the acceptance, understanding of the public of the dashboard, and as well as its use, whether you were tracking this. Do you have information on that, and how far the public actually looked at this and maybe changed its behavior or adapted based on the data that were provided?
I don't have great information, unfortunately, and it would be very interesting and useful to have some. We're sort of in a very, very long, drawn out process of moving the hosting of our dashboard, trying to bring it internally where I think we'll have more ability to gather some of that type of information, but I mean, we're going on 7 or 8 months now and it hasn't quite happened yet. Still waiting for the infrastructure folks to do their infrastructure things. I think in general, we have spent a fair bit of time trying to make it reasonably digestible to folks, especially on the public side. On the more internal side, I have more interaction with those people, and so I often will hear more questions. And you can, of course, when we have a dashboard that's restricted to public health practitioners, you can reasonably expect a certain level of public health knowledge. Hopefully they actually have that level of knowledge. With the public, you know, I think it's important to really lay out and present the information in a way that leads them naturally to the conclusions that you feel are valid. That is the point of presenting these things. It's not just to present, here's a bunch of data, make of it what you will. That's not helpful to the public. They will, you know, all too often either get the wrong idea or just say like, I don't have time or interest or, you know, level of the amount of effort needed to parse all of this, so I'm just going to ignore it. Part of our job is to present the information in a way that we feel is the most valid and the most helpful to protect their public health. And so we do go to some effort to try to accomplish that with dashboards and try to make them easy to understand, easy to digest. But there's, of course, always room for improvement.
We all suffer kind of from first to having to learn and then to understand what people see when they look at our data, because
yeah, it's sometimes not the same thing as we see, that's for sure.
But I have another question for the dashboard. I think when you flashed the data, it looked as if you were normalizing by millions of people because you essentially covered the entire state. When you showed us your case data, I noticed you used Logan. I think the population is about 50,000, and then the other kind of success was on the order of 9,000 people. Mm-hmm. There was a paper early in May of 2020 going through different scenarios and acknowledging that the virus decays during travel in the sewer pipe. So that in essence, the actual reach of what we see is much more limited than the wastewater that flows in from regions. And I think as long as we have a homogeneous distribution of cases across the sewer shed, it doesn't really matter. But as we know, and I don't know the stratification of the population in Utah, but I would assume that the behavior, income, people, you know, all kinds of factors vary. So have you done an analysis of how far the actual reach of your wastewater network is, and maybe then normalizing to that closer, narrower range and maybe getting better correlations?
No, we haven't. It's a great idea. I think it'd be extremely useful information. We haven't done, I don't know, maybe some people have, which would be awesome. I'd love to hear about it. I think you'd have to do it. I think there's so many factors that are different from facility to facility that you probably have to do it for each and every one that you're doing. Possibly by season as well. We get some big differences, not just in temperature, but also in the infiltration of runoff and groundwater season to season in some of our facilities. You can have some of them are very, very leaky. Yeah. And they'll pick up a lot of groundwater and runoff, and sometimes a lot of agricultural runoff as well.
Yeah.
And I absolutely agree. I think those points you made probably, at least in some scenarios, possibly all the scenarios, have a major effect on what we see. And we have some very large, some of our sewer sheds are very small and cover small populations. And I agree, it's probably not as important there, but some of them, our largest one covers.
We all suffer from these limitations, right? Looking at the clock here, I think we're running out. So let's head over to the next presenter.
Great question.
Thank you much, Nathan, for a fascinating talk and doing this early work. It's much appreciated. Back to Ellie.
Okay, Loren, are you ready to share your screen? Thank you.
Okay, sorry. Hi, I'm Loren Hopkins. I'm a Chief Environmental Science Officer for the Houston Health Department. And I'm going to talk about just how we use the wastewater in public health interventions here in Houston. Before I get started, I want to recognize this is a collaborative project. It couldn't happen without our partners. The Health Department and Houston Water worked really closely and continues to work with Lauren Stadler and Kathy Inzer and the team from Rice University, as well as in the beginning, we worked, really set up, Rice worked with us as well as Baylor College of Medicine virology experts. We continue working with Rice University and do the analysis that Baylor was doing early on for us, we're doing at the Houston Health Department Laboratory. I also want to just, quickly acknowledge the rest of the professionals, and I think the list is probably even longer than this, but just to flash the name up of, how intense the collaboration is to make this project continue to work. Really quickly, this is our process overview. We take 24-hour influent wastewater samples from 39 wastewater treatment plants here in Houston. We always joke that Houston Water is so frustrated that we have so many wastewater treatment plants for our sprawling city, but when it comes to wastewater surveillance, we love it. So we have 39 wastewater treatment plants that cover 2.3 million plus people. Houston is the 4th largest city in the US. We also do a lot of manhole sampling weekly, schools, congregate living facilities. And because our wastewater treatment plants, sewer sheds are different sizes, and some of them are much larger than others, we also sample at lift stations to try to get a little bit of breakdown in some of those big areas. Those samples, as I mentioned, they go to 2 different laboratories every week, Rice Baylor, and then now Rice Health Department. The Health Department and Rice use statistical models to assess the data in different ways. And then what I'm going to be talking about here is this Health Department use of that data. All right. And just to give you a really quick look at it, this is what we're talking about. The big sections, of course, are the wastewater treatment plants, and the dots on here are the lift stations and the manhole sampling. So a big logistics effort all week long by the team. All the samples are collected by the city itself. I think what may set us apart differently from other programs is that it is run out of the health department and in partnership with Public Works. Okay, so in terms of intervention, I think I broke it down into about 5 different steps here, which I'm going to hit on. The first one is obviously super important. It is the wastewater surveillance reports. That's where all the data from the wastewater that we get in the weekly samples is analyzed and comes together. So we create this weekly report with information evaluated at the sewer shed level, the zip code level, and the manhole level. And then that information is communicated to leadership via email and in a leadership meeting. And that email, it's kind of like I'm talking to you about how to write an email, but it's actually really important because this is intense, a lot of data. And obviously, listening to Nathan, listening to the discussion, most of the people I'm talking to or that we're relaying our information to are not big wastewater people. There are people that are going to use the wastewater information. So we provide it, very carefully to them. And we've learned that we've been doing this, for a very long time, it feels like. And so we figured out, how to best get the information across. So what we do is we email them and they get this intense report attached based on, what the sewer sheds are doing, what the zip codes are doing, and what the manholes are doing. And then we put key highlights in the body of the message so that they can just see, okay, this week I don't have time to really look into it. So I'm just going to look at the key highlights and a quick look at the manhole analysis so they can see what manholes are popping up as having high positives. And that leadership that I'm talking about here consists of the public health authority, the director of the health department, the assistant directors of the health department. And then our public health authority loves wastewater, and he thinks it's just the best thing because when we were in the pandemic, this is one of the places where he could really feel good with the data because, as Nathan said, there were issues with testing and things like that through time. And so the public health authority, he communicates the information to the mayor and to the medical center leadership. We have a very big medical center here in Houston called the Texas Medical Center. It's almost like a city unto itself. And also infectious disease doctors. And then he also conveys it to the press when it's needed. So in terms of that medical leadership, they actually discuss the wastewater on a call with all the heads of the hospitals. And they look at the viral load and the positivity rate against what they're seeing in the hospitals. And they, just to give you an example, they suggested, I was listening to them talk about using the wastewater sewer shed map to actually inform where to allocate surge staff nursing. And I think, again, back to what Nathan said, when we're able to use wastewater as a predictive tool, they're using it as a predictive tool. So that was very helpful to them. We also convey that information on the program manager level. It's discussed in a meeting called the Data to Action meeting that's held every other week. And the purpose of that meeting is just to bring together the leads from all the aspects of the different interventions that are going on. And that's the lead from the school strike teams, the nursing home strike teams, the congregate living, the jail, the vaccine, the outreach, testing, and reporting strike teams. So that's about a 2-hour meeting, and the wastewater is part of that meeting. So the wastewater report actually consists of temporal trends. I'm showing you one right now of the viral load estimate, and then of it compared to the positivity rate, and broken down right here. We're looking at it by sewer shed, and we also look at it by zip code. So that's sewer shed, all the zip codes within a sewer shed. We look at the actual, what the positivity rate is in the zip code and what the viral load is in the sewer shed, in the zip code, based on the sewer shed information. We provide heat maps and we benchmark our information because in terms of the health department, their understanding is our first peak, which was, and when we started getting really good wastewater data, when we kind of got out of our pilot phase, was July 6th. So everything's benchmarked to that peak. And so, as we go through time, the question would be, is it as bad as July 6th? Was it bad as our first peak? And so that's how we measure things. And it's been a lot more helpful to communicate that way. People, within the health department or necessarily get their head around the viral load by itself. So that's how we do that. So you're looking at those two maps, and so those reports come out every week. Like I said, this is a look at what is in the key highlights that we found to be important. As I said, it's been massaged through time. We have a total indicator. So on October 11th, you can see that the viral load across the city was 184% of the July 6th level. And the week before that, it was 130% of the July 6th level. And then we talk about which wastewater treatment plant is increasing or decreasing and how many are increasing and decreasing. And you know, how many zip codes are increasing and decreasing, things like that. So it's just a summary of that information. And how many people are being affected. And you can see an example of something that's in the report and in the email that comes out. The blue line here on this graph is the positivity rate, and the green line is the wastewater viral load. More material that goes into the report. This is one of our heat maps. This is an example of one that is what is listed here is every wastewater treatment plant and the total across the city. So that you can kind of get a sense of, like, you know, are we coming out of a peak? Are we going into something? You know, when is it calm? When is it not? And where it's happening. So that's just like an easy way for them to look at it. We make lots of these kind of heat maps because we find that it's very simple for them to understand. Here, you know, we provide our variant data in terms of heat maps. This is just an example. I didn't want to provide you with all of them, but this is for the Delta. And you can see that when we provide it, we also indicate what zip code of zip codes that I'm going to talk about in a minute, about how we identify what zip codes are important to us, whether the variant is in that zip code or not. Okay, so we don't analyze the wastewater data by itself. We've already talked about the positivity rate, but we really pretty much give every bit of information we can in the health department. We bring it all together. So it's a multi-data analysis to help us direct where the intervention should go. And so really quickly, you know, here's an example of how we analyze the testing rate that was discussed earlier. We look to see what zip codes, where they're standing with their testing rate, and the number of positive cases per person. And that's on the left, and on the right is just another piece of information, how the vaccination rate, the rate of the vaccination rate and the fully vaccinated rate, and the positivity rate look at a zip code level. And that will indicate to us, you know, how is the testing in a zip code? How is the, you know, where are we low vaccinated and high positivity rates? So that's the kind of information that we put together with the wastewater data. And then we analyze it for that data collectively for the priority zip codes. So the priority zip codes are ones we're going to do targeted intervention. In the beginning, it was health education, it was in testing, and now it's vaccine and continued testing. So they are, you know, we obviously work across the city, but we do target areas that seem to be more heavily affected. And that comes from this set of data. So priority zip codes are really, first of all, there's 105 zip codes across the city, and we look at these COVID community building index, and those are tier 1 and tier 2 zip codes. So generally the targeted interventions are going to be in those zip codes. So we take that subset, and then we look at what's in those zip codes in terms of the positivity rate, the 5-week positivity rate, the unvaccinated rate, and then we compare that with the wastewater data, what the trend is in the wastewater for that zip code. So that's one method of getting priority zip codes. We'll take the worst from that blue category. And then we also take the worst from the priority zip code herd immunity calculation, which is also based on the wastewater. And not just the wastewater, but it has wastewater in it. And then that's how we determine where we're going to put special pop-up sites or special door knocking campaigns, faith-based organization, community-based organization initiatives. So with the email with the report, we also provide an Excel file that they can sort if they're interested in it. And so, you know, some people will just read that email and some people will actually get into the data and analyze it. It depends on what group you're in and what you're interested in looking, you know, to see, you know, whether maybe your intervention is being effective or not. So this is the kind of thing that is in the information that goes out. Once we've identified those priority zip codes, we then really do a deep dive in that and summarize the material specifically for the strike teams or our leadership so that we can follow how we're doing there. And this is what comes out weekly for the zip codes that we have seriously prioritized. The vaccination rate, and in parentheses, whether it's going up or down since the previous week, the wastewater trend, the positivity rate, on this plot, you can see the blue line is the positivity rate, the green is the vaccination rate, and the red is the wastewater. And, you know, of course, those two little dots there are the future prediction of the wastewater. We also do a network analysis within that zip code and say, okay, what's a good place to put, you know, a vaccination site based on like people walking with a half mile. We actually even provide more information that I'm going to show you. We break down who's not vaccinated, what, you know, what age groups and what their racial breakdown is if not vaccinated. We calculate how many people need to be vaccinated to get to 80% vaccination. And we let the zip code know how many people they need to really have a protected zip code, because we're searching for the 80% level everywhere. And kind of like pledging, this is how many you need to get vaccinated to reach your limit. This is just a slide of how the herd immunity analysis works. This stuff in the box here, these zip codes, those were the ones that were chosen as the high priority because they have the highest PCR positive per 1,000 people. The other analysis is on positivity rate, but we use two kinds of analysis. And then this is the rate of antibody protection. And the herd immunity is just the wastewater, you know, it uses lots of different information, including the wastewater data and some information from a seroprevalence study that we had done here in Houston. And then our vaccine database and our disease surveillance database for PCR. And then we provide interventions, and just, you know, for the manhole data, that's one that I didn't hit. We right now just divide the concentrations into high positive, low positives, inconclusive, and negative. We provide that information to schools. We provide it to our school nursing team. And we, this right here is a heat map showing the nursing homes and the shelters and where they stand. And then, you know, they'll follow our, you know, our group will follow, our health department contacts the schools and the nursing homes and wherever, and works on outbreaks and trying to understand why there are positives in the waste. And we have a standard operating procedure for that. And I guess just the last thing is we do, you know, the public health authority, this is him talking here on the right. We have a, you know, we're tweeting about, we're educating our public about how to understand it and, you know, what it means to them, and just using it as a tool really right now and just pushing people more and more to get vaccinated. And then we also have a dashboard, and that dashboard on it includes the viral load, the positivity rate. You can look at it for your zip code, and the unvaccinated rate. So we're really trying to get people to focus on, you know, hey, you've got a viral load, you've got a high viral load in your zip code, you know, your positivity rate, in this case is 8%, it's not very high, but, and the unvaccinated rate, just to really try to connect everything together, that if you don't have anything in the wastewater, if you have something in the wastewater, you want to be vaccinated, and really just trying to get that messaging out there. And I think that was my last slide.
Wow, that is incredibly impressive. Thank you so much. We have a couple questions, and I have some of my own, so I'll just rapid fire through these, and reminder to everybody to put your questions in the Q&A box. So The reporting is just very impressive and interesting. So one question is just how much time is involved, staff time is involved in making these reports? How much of it is automated? It looks amazing, but it looks very time intensive if it's not automated.
I mean, so that work, so one of our partners is Kathy Enzer here in the stat department. I'm actually also in the stat department, and she works with one person at the health department. So we have teams that work like that. And so I know that eventually Dr. Enzer wants to, let Rebecca with the health department, she has, a master's in statistics over there and, take it on, but there's still like little tweaking things that have to happen with the data. Quality of the data is so important to me. I run, like I said, I run this program, and it'd be very, very stressful if I wasn't super confident in the data. So that's why we send our data to 2 labs. Every lab does replicates. The statistical model does the between lab and within lab variation calculations. So all of that has been going on for a long time, but I believe that, it's less and less effort, and we actually used to do more, but so we have one wastewater statistician that can analyze all of that. But then when we add in the positivity rate, the vaccine rate, the unvaccinated, the herd immunity, in the data science group at the health department, which I also run, we have maybe 3 people that work on that. Awesome. But in the pandemic, it's really, really important. So yeah.
I guess on that note, because you've done a really interesting job integrating the wastewater data into all your other health department data measures, one question is, how do you see this continuing in the long term? Maybe other targets? It seems like you have leadership that's really bought into wastewater. And so how do you see this going forward?
Right. Well, one thing is we really need to, as a group, all of us, I believe, really prove that this is something that needs to be continually funded. So health departments are funded from the CDC. A lot of funding comes through that. Many of us on this call, I'm sure, have CDC funding for the news and things like that. But we're using Dr. Stadler is analyzing for flu right now for across the city and for schools, and we're trying to understand if that data is actually going to be that useful. Right. So and then we are looking at other things, and our intent, when we have what is like 100 and something samplers out there, we've invested heavily in the city's surveillance program. But, yes, it's time that we ramp up and prove that, it can be used for all kinds of different things. I mean, we're hopeful. Yeah.
Amazing. So have you seen, since you have this resolved data on vaccinations, have you seen an impact of vaccination on wastewater concentrations? I guess this is probably trying to get at with your data, if you have any understanding of, I guess it's so difficult right now, but shedding varying with vaccinated individuals that have breakthrough cases. Oh. I'm assuming that's what that question's trying to get at, or maybe I'm just adding to it.
With the Delta variant coming on, maybe it's not the same number of people that we're tracking. We understand that, and we've been we're doing a lot of work on we do breakthrough analysis here. We do reinfection. We don't have a good handle on it, no, but we understand that we need to have that in our herd immunity model. We need to refine those variables as we go forward, as we can get more information. And when, I don't know what the question actually, but for our nursing homes, we're sampling, we sampled before they were vaccinated, we sample after they're vaccinated, we track everything. We could see nothing in nursing homes now that are completely vaccinated, yeah.
Okay. I guess this is a very practical question, very in the weeds question, but it's interesting to me because most health department data is at a zip code level. And unfortunately, we don't make our sewer sheds perfectly at zip code level data. And so I'm curious how you talked about presenting wastewater at the zip code level. And so I'm curious how that happens? Are you like aggregating different types of wastewater, or do the lift stations give you enough resolution for you to be able to disaggregate certain zip codes?
You know, I think that's a doctor answer question, but basically we know how much of a zip code is within each wastewater treatment plant. And then there is a weighting if, you know, if a zip code is completely split between two watersheds or sewer sheds, then, you know, it's an estimate. But we do have positivity rate. We geocode everything in the city. So we have positivity rate perfectly by the zip code. And then when we add it in, and then the relationship looks really good. So we're thinking, you know, we're thinking that it works. But cool. That is the point of doing the lift stations. We have 105 pumps. We have 105 zip codes. We really wanted to do, you know, lift station level, more granular level information, which we are working on right now.
Cool. And then on the positivity rate question, someone in their community seeing positivity rate affected by testing behavior, and then areas of this or cities where the wastewater suggests there's under testing happening. And I guess curious how you've observed, you know, testing penetration being affected, and whether or not you can see that in the wastewater data.
Well, you know, I mean, maybe that's something that I should have brought out really clearly, but there are areas of town, there's certain communities that really are just not taking up the testing. And way back when we wanted everyone to test, you know, we were really pushing to get everybody to a certain low percentage, you know, a baseline of testing. There, you know, certain zip codes, you know, were lagging. But the great thing about it is that once we really understood what our wastewater was doing and we were confident in that data, then we're really kind of relaxed about the testing now, because we know how well it's representative. That's what the public health authority is so interested in, you know, instead of, you know, which is I think it had Nathan's slide. The good thing is that, you know, it's an unbiased sample. So once we were really confident in our data, we're good. And that, I think, is the beauty of it.
Amazing. Okay, let's do one last question before I turn it over, and that's, I guess, what are the challenges with sample collection fatigue with the city workers? Is the city wastewater utility the one collecting all the community samples as well as the samples at the wastewater treatment plant? Who's doing the actual collection?
They're health department employees. They're in my group. I think we have 4 or 5 of them. Well, I think we have 4 and a manager, and they're awesome. I mean, you know, they're overqualified. They just wanted to do it in the beginning. And I think they are tired. Yeah. So we would probably have to switch them around, but like, no, Public Works here couldn't take it on. It's massive, you know, but we have a great working relationship with them, and they're very supportive of it. So no, you just have to hire people specifically for that.
Yeah. Okay, well, I'm sure everybody wishes we could poke your brain for a really long time. Thank you for the presentation. Thank you for coming. I think Jill had a couple announcements, so I wanted to pass it off to her.
Yeah, thanks. I know we're right at time, so we'll close here in just a second, but I wanted to post a couple links in the chat. We are having our RCN fall meeting next week, November 3rd. There's a great lineup of presenters, so we hope you all will be able to join us for that. And then I also included our link to our upcoming webinars. We have one more webinar scheduled for next month, coming up in a few weeks. So with that, I think we're all set. So I'll say one more thank you to our presenters, Loren and Nathan. Thank you both so much for spending the time to talk with us and sharing your perspective on all of this. We really appreciate it, and hope to see you all again soon. Thank you.